Well, hello, everybody. Look who's here. Look who's here. Cora Stone is here. Yay. Life is good. Wow. How many people are here? How many people are in this damn class anyway? We got 34 people here. I don't know if I think 48 or 50. Jack. Go beat myself. Robert Johnson. Can I see all your faces for a second? Just so I can say hello. Billy Corley, Willie Corley. Everybody's up. Megan Jordan, my son, Sydney, Stefan McCain, Yusuf, Julia Waters. Is that Matt Gremlin? Is it Gremlin?
You know my name.
Yeah, I'm an idiot. Okay. Looks like we got everybody up. Life is good. Should we get started? I think this lecture might be less than an hour and 15 minutes. I'll have to apologize if it is. I know you like to spend as much time with me as possible. Shall we begin? Let's, okay. So, I'm gonna bring up my notes. Oops, hang on. Okay, bringing up my notes here. I'm gonna, oh wait, let me bring up my chat screen too. So if you need to chat, if you have a question and you don't want to talk, you can just speak up anytime you want, but if you want to talk, go ahead and chat and I will see it. Stefan McCain, I love your name. I'm sorry, but it's just the way it is. Okay, so we finished up last time talking about routes of administration. Got through them all, and now we're moving on to the next section, which is distribution. So distribution, I believe a lot of you are sophomores, if not juniors. Hopefully you've had a little bit of anatomy, like so you know the cardiovascular system, but we're going to go through it a little bit, just to make sure everybody knows what we're talking about here. Let me pull up my figure. Oh, Slish, you've got a lovely figure. Well, thank you. There it is. Okay, so the cardiovascular system has got two circuits. You've got the systemic circuit and the pulmonary circuit. So in this figure, the systemic circuit is both the bottom and the top of this. Let me ask a quick question. Matt Gamelin.
Yep.
I can see your face. Can you see my arrow when I'm moving around on the screen here?
Yeah, I can.
Okay, good. Thank you. Okay, so your systemic circuit then comes out of the left side of the heart, and it splits. Part of it goes up to your upper body, I guess arms and head. I don't know anything about anatomy. Why am I even pretending? And then the bottom, I don't, you know, what the hell? Anatomy, this is an arm, this is a head. That's my anatomy. Okay, I'm a cell biologist. Okay, so the bottom part of it comes down here, goes to your intestines, the whole rest of your body, and then the lower circulation. It all comes back into the vena cava, goes into the right side of the heart. From the right side, it goes out to the lungs, and this is your pulmonary circulation, comes through the lungs and back to the left side of the heart. So you've got arteries that lead away from the heart. Arteries get smaller and they become arterioles. The arterioles get smaller and smaller until you end up with these one cell thick vessels called capillaries. So in the capillaries is where you get exchange of gases, exchange of chemicals. So you can see here that in this loop here, you get a capillary bed here that goes through the intestines, and that's how things are absorbed from the intestines to the capillaries. And now here, this is neither an artery nor a vein. This is a portal system here. So it goes from the intestines to the liver. So portal means other than just straight from arteries to veins and back to the heart, there's something between two different organs going on. So these capillaries then fuse up and then make larger vessels, and those vessels then make their way to the liver. Now you've got a second capillary bed here. And then this is what I was talking about last time with the first pass effect. Everything from your intestines goes to your liver. Now your liver will metabolize it or not, and then put it back into the venous side, and it comes back to the heart. And another thing I was talking about last time was how inhaled drugs go in even faster than injected drugs. When you inject a drug, 99.999% of the time you're injecting into a vein. So from the vein, that drug will come back to the heart, and then from the heart, it goes out to the lungs, and from the lungs back to the heart, and from the heart, then it can go up to the brain. When you inhale something, it goes from the lungs to the heart and up to the brain. So it's an even faster route than injection. If you inject something subcutaneously, it has to go through the capillary system, into the capillary system, and then into the venous and back. So that's even slower than IV. So SC or IM would be a little bit slower than IV. Okay, so that's your circulatory system. I don't want to spend a lot of time here because I think it's kind of on the simple side. And like everything else, polarity affects distribution through the blood, through the circulatory system. If you've got a polar drug, it will go into the blood system pretty easily, travel around in the blood. If it's non-polar, it's not really going to dissolve in that water system of the blood. What happens is non-polar drugs will stick to proteins called albumins. So there are albumins in the blood that help to carry non-polar things around. There are also, high density and low density lipoproteins, which are just little balls of fat, and that helps to distribute non-polar drugs as well. And let me check my notes again real quick before we move on. Make sure I'm not forgetting something. Capillaries, veins. Okay, so two-compartment model. Again, I briefly mentioned this last time, so here's where we really want to talk about it, because it'll come into play a lot with all our drugs. So your body really isn't made of two compartments. It's kind of two theoretical compartments. One is the water compartment, which is cytoplasm inside your cells. The blood is mostly water. Interstitial fluid is water. So those are your water compartments. Then you've got your lipid compartments, which include all your cell membranes. What else? Subcutaneous fat between your organs. There's a lot of fat between your organs, padding them. Things like that are going to all be your lipid compartment. And keep in mind, your brain is pretty fatty, right? Your brain is all neurons, and neurons are mostly membrane. So your brain is very fatty. So if anybody calls you a fathead, thank them, because it's a compliment. It means you got a lot of brains. So the average female, of course, has got a higher body fat percentage than the male. So they've got a larger fat compartment than men do. But anyway, when a drug goes into your body, typically it's starting out in the water compartment. You're either going to absorb it through your intestines and it goes into the blood, or you're injecting it into the blood, you're inhaling it into the blood. It's going to start out in your water compartment. As it gets distributed around the body, if it's more soluble in fat than water, it's going to distribute itself into the fatty compartment. And then it's an equilibrium system. So it's not like it's just going to go there and stay, but it'll go there and slowly come out. So it'll come in and out and in and out. Most of it will be here. So the way you want to think about that is, if it is, say, if it's distribution, what is it? Coefficient of, what is it called? oh God, I forget what it's called now, but the distribution between fat and water. Say it's 90% fatty and 10% is in the water, So you take that vessel I talked about last time where you've got like a fat octanol basically in water, you shake it up, 90% of it's in the fat, 10% of it's in the water. So at equilibrium, 90% is here and 10% is here. So imagine you took 100 milligrams of something. 90 milligrams are here, 10 milligrams are here. Now, elimination only happens from the water compartment. We'll talk about that more today. So as you lose, say, 1 milligram from here, another milligram is going to come out and replace that. So now you've got, 99 and 1, or you had 90 and 10. Now you've got still 10, but now you're down to 89. So it's like it'll always be in equilibrium between the two. So as it's slowly coming out this way, it'll slowly come out of the fat. So when you take a very fat-soluble drug, it'll partition into that fatty compartment, and then as the little bit that's left in the aqueous comes out, it'll come out of the fat and replace that aqueous, so you get this slow elimination. If something is only in the water compartment or mainly in the water compartment, it'll come out really quickly because that's where the elimination happens, right there from the water compartment. And also based on the kidney function that we'll talk about later. So you take something that's very water soluble, like vitamin C, very water soluble drug. You can try this if you want. You can get these 500 milligram vitamin C tablets, like, oh, good, I've got my whole day's worth of vitamin C right here. And you chew that pill and you swallow it. Half an hour later, you take a pee, there's your vitamin C. It all just comes right back out because anything water soluble gets washed out of your system pretty quick. Okay, that's our two compartment model. Again, it's not two physical compartments, but it's two theoretical compartments. The drug can either will be in both really, but it'll be majorly in one or the other depending on its own polarity. And this will be really important when we talk about elimination of fatty soluble drugs. Okay, blood-brain barrier, our next topic. Let me make sure. I just want to keep up with my notes to make sure I'm not missing anything. Blood-brain barrier. Okay, so getting back to our distribution, out in the body, like most of your circulation is your systemic circulation, and the capillaries in your systemic circulation are like this. They're the walls of the capillaries, if you look over here, are just one cell thick of endothelial cells. And these are very flat cells. They're basically interior thin cells, endothelial. So these are very thin, and they've got gaps between them, fenestrations, I believe they're called. So if you've got the blood that's in these capillaries, because it's being pumped by the heart, the pressure coming into the capillaries is high, and that forces fluid out of the capillaries. So anything dissolved in your capillaries will leak out into your tissues through those fenestrations. These are intercellular gaps, so you got this fluid leak. And then it's not like you keep swelling up over time. What happens is your lymphatic system will pick up the excess fluid and take it back and run it through the lymph nodes, make sure there's no infections there, and then send that back into your bloodstream and into the venous side. And this is why if you've got a cold, they say drink a lot of fluids, right? They say, what do you do if you have a cold? Well, no drugs kill the cold virus, so drink a lot of fluids. And if you drink a lot of fluids, that increases your blood pressure, increases the pressure going into your capillaries, causes more fluid to leak out of them. That fluid then gets picked up into the lymph glands, and then your immune system can take care of any viruses that get picked up through that system. So you're kind of like flushing viruses into your lymph system, and they're interacting with the immune system and taking care of it. So if you've got a virus, it's good to drink a lot of fluids. But anyway, that's most of your body. The only place that's different from that is in your brain. Oops. Oh, here we are. Capillaries in the periphery, blah, blah, blah. The only place that's different is in your brain, and this is what we call the blood-brain barrier. You've got glial cells there called astrocytes. Glial cells are kind of helper cells in the brain. We used to say they don't do any signal transduction. Turns out they do a little bit, but we don't get into that. But anyway, they're like support cells in your brain, keeping the neurons happy. Another thing they do is they send these little feet out that wrap around the capillaries. So these feet are basically just layers of membrane. So you lap layers of lipid bilayer around the capillaries, and that blocks these, and there's no fenestrations, there's no leaking out of the capillaries. So now you've got a really tight seal between the capillaries and the blood inside and the interstitial fluid that surrounds the brain. So if you've got some kind of a polar toxin, it really can't get out of the capillary and into your brain. It's a way of protecting your brain from substances. If something is nonpolar, it can diffuse right through lipid bilayers, so it will get into your brain. So blood-brain barrier, the only way something can get into your brain is if it's nonpolar. And so all the drugs that we talk about, the ones that affect your brain, will be nonpolar drugs. And again, we'll talk, when we get into opiates, we'll talk about how, nothing's totally polar versus totally nonpolar. It's all on a range. So something like morphine is not very polar. I mean, not very nonpolar, kind of more polar, but it will slowly get into the brain by kind of leaching into the brain over time. Heroin's a lot more nonpolar, so it gets into your brain faster. Fentanyl is very nonpolar, boom, goes into your brain really fast. So the ability of something to get into your brain is based on its polarity, and that's because of this blood-brain barrier. Okay, let's stop share for a second. Let me get my chat up, get our chat on. Oops. Trying to get a little discussion going here, Matthew Cole, Mr. Quiet. Mr. I don't want to talk to Dr. Slish because I don't like him. All right, let's go back to the notes here. So that's our blood-brain barrier, specialized capillaries that protect your brain, keep substances out. Lipids. Oh, okay, so the other thing I want to talk about here is two areas in your brain that don't have this blood-brain barrier. One is called the chemical trigger zone. So this is in your medulla. Did we chat? Someone's chatting me. So is there a major difference between the two compartment model in regards to sex? No, there's not a difference in how it works. There's just a difference in the amount of the lipid compartment versus the aqueous compartment, okay? And again, that's average. I mean, if you got an overweight man and a thin woman, it changes that. And there's really no functional difference. It's just a difference of degree of how much lipid compartment there is. Okay, chemical trigger zone. So your medulla is like one of the, so the medulla is, it's the first part of your brain above the brainstem, and it's involved in a lot of, it's all automatic stuff. It's nothing you ever know about or have to deal with. It keeps your heart rate, your blood pressure, all the housekeeping things go on there. That's where you've got this chemical trigger zone. And what happens here is if you've got toxins in your body, it'll activate your chemical trigger zone, and then you'll throw up. So the way you want to think about this is you eat something that's toxic, it gets absorbed in your small intestine, gets its way through the liver somehow, makes it to your brain. Your brain says, wait a minute, what the hell did you just eat? And you start throwing it up. So you've still got some in your stomach, and you're getting rid of it, protecting yourself. So that's your chemical trigger zone. And this is what causes overdose in some drugs, because, opiates especially will slow down your respiration and activate your chemical trigger zone. So you end up vomiting and then breathing it back in, and you choke on your own vomit, they say. But basically, it's called aspiration. You aspirate your vomit, and that blocks your airway, and you die from that. So alcohol, opiates, those things tend to kill you this way. So that's your chemical trigger zone. The other place, which is not that important for us because we don't get into this, is in the hypothalamus. So the hypothalamus then releases hormone-releasing hormones. ACTH, adrenal corticotropin hormone, gets released here. It goes to your adrenal cortex, and your cortex then releases hormones based on that. So it's like it's the way your brain has of releasing hormones that affect the rest of the body. So it needs to have that open access so it can secrete things. Not that important here. Main thing I want you to remember in terms of blood-brain barrier is it protects your brain from everything polar. And the only place that doesn't work is in your chemical trigger zone, which is there to protect you from toxins. In terms of placental barrier, there's really not much of a placental barrier. Fetal capillaries come in contact with maternal blood. If anyone had me for developmental biology last spring, we talked about this. Basically, maternal blood just gets released into this little area that bathes the maternal or the fetal capillaries, and then it gets sucked back up into other vessels and goes back into the maternal venous supply. So you've got basically very little barrier there between maternal blood and the fetal blood. So basically, if you're pregnant, any drugs you're taking, your fetus will be taking as well. And that's where we get into teratogenicity. So teratogenicity means birth defects. Okay, so that's it for distribution. Hang on a second. Matthew Cole, what does charcoal or ipecac do to induce vomiting? Does it trigger the medulla? So not charcoal. Charcoal basically it binds up organic chemicals. So they have you swallow charcoal, and if there's anything left in your stomach or intestines, it will bind to that charcoal and then pass right on the other side. Ipecac does directly activate your medulla and causes you to throw up. Usually when I do this in person, I show this animation clip from The Family Guy. It's where like everyone's sitting around, Stewie, the dog Brian, and the father and the older son, the stupid one. They're all sitting around. They have a contest to see who can drink ipecac and not throw up. And it's just hilarious because they all just barf all over the place. If you like barf jokes, it's funny anyway. Yeah, you should look that up because it's pretty funny. It might even be in my Moodle page. Absorbs chemicals. Okay, so Christy answered that question. Induces vomiting so that someone who hasn't taken a chance can be absorbed. If morphine is inhaled, does it leach into your brain faster than if it was injected? A little bit, probably. That's definitely true for like a fentanyl. If you can inhale it, usually they don't inhale fentanyl. But because morphine is kind of on the slow side, inhaling it doesn't make it happen that much faster than injecting it. The time scale is about the same. Yeah, so not that much faster, but it can get into your brain through that way as well. Okay, any other questions, comments? Okay, so if morphine is found, I need to write. Thank you. All right, so metabolism. So I wrote this in your book. The thing about that hepatic portal vein, I guess it's called. The reason for that is kind of like an evolutionary mechanism to protect you from yourself. We eat a lot of stupid stuff. But if you think about in terms of evolution, animals eat plants to live, and some plants don't want to be eaten. So they've developed, through evolution over time, developed toxins. So the animal eats the toxin, gets sick. And some animals then, you've got a variation of population. Some animals will not get sick, and they'll pass those genes on. The reason they're not getting sick is because their liver breaks down that toxin. And then maybe the plants get even more toxic toxins. So there's always this back and forth evolution, over the, hundreds of millions of years, I guess, of evolution, that liver enzymes are trying to break down plant toxins. Plant toxins are trying to get animals to stop eating them. So, that's why we have these liver enzymes, and that's why everything you eat goes to the liver first to clean it up. Some things plants make, they want you to eat, right? Fruits have got their indigestible seeds inside this tasty package. So animals eat it, and then they drop the seeds off in a nice little pile of fertilizer. But other plants don't want to be eaten, and so they've got toxins in them. So, role of the liver: produces digestive bile to absorb fats. I think I talked about that. And the key to the liver metabolism is what the liver is mainly trying to do is make toxins more water soluble. Two things about that. One is if it's water soluble, it can't get into your brain. So it's protecting your brain by turning it water soluble. And the other thing is your kidneys can only get rid of water soluble things. I'll talk about that in a minute when we get into elimination. But the enzymes in your liver, there's a series of enzymes called the cytochrome P450s. And they're not doing something specific. It's like most enzymes in your body or in your cells, like glucose phosphorylase, is going to take phosphorylated glucose and take the phosphate off. Boom, that's it. That's all it does. It only has one substrate, and it produces one product, okay? Most enzymes are like that. These cytochrome P450s are more generalists, so they're going to grab any organic chemical and change the functional group on it and let it go, So that's, and the generalness is because your liver doesn't necessarily know what chemicals are going to be coming at it. So it's doing general things to mess them up. Usually you're going to make that toxin more water soluble and get rid of it. Sometimes you activate that toxin because, it's not specific. One example I give here is psilocybin, right? The magic mushrooms. The drug in the mushroom is water soluble, so it can't get into your brain. But when it goes to your liver turns it into psilocin, which is nonpolar. Then it gets into your brain. So it usually inactivates things. Sometimes it activates things. Okay, so metabolism, mainly about making things more water soluble so you can get rid of them. First pass effect. Anything absorbed in the intestines passes through the liver first. Many drugs are broken down before they reach the bloodstream. Now you can get over that clinically if you know, oh wow. I just got this big yellow line across my notes. Did somebody do that? That's kind of cool. Anyway, oh, somebody's doing this to me. Who's doing this? That's a lot of fun. Okay, so you could, if you know a drug is rapidly metabolized, you could give more of it to get it in, but usually you would give it by a different route, injection or something. Okay, enzyme induction and inhibition. This is kind of important too. So, like every single protein in your body, it's going to be regulated. The amount of it in your cells will be regulated. So there are some cytochrome P450s that will be strongly upregulated by certain drugs. And there are some, and so like, I think one of the classic examples is phenobarbital. Phenobarbital is a barbiturate. It causes depression, not emotional depression, physical depression, slows you down, tires you out, just like Valium. But phenobarbital induces your liver enzymes. So you'll get this metabolic tolerance where the more phenobarbital you take, the less effective it is because the enzymes come up and it gets broken down faster. Another aspect of that is kind of like a crosstalk, where if phenobarbital increases the enzymes that are working on phenobarbital, but also for other drugs, then the other drugs get broken down faster too. So you get this crosstalk between chemicals where, you know, one drug increases the number of cytochrome P450 enzymes, and the other drug goes down in concentration in your blood. But bottom line being, your liver enzymes can be induced, so you get more and more of them over time by drugs. They can also be inhibited. Your liver enzymes can be inhibited. Typically, this is not like a down regulation of the proteins, but like a physical inhibition of the enzymes themselves. So the best example of this is when people drink something with a lot of phytochemicals in it. Phytochemicals are things that the liver enzymes would want to break down. So a classic example is people taking their drugs with grapefruit juice. Grapefruit juice has got a lot of phytochemicals in it. So, you know, you take your drug and you take your grapefruit juice. The grapefruit juice blocks all your liver enzymes because there's just so much phytochemical in there. All the enzymes get busy. So the drug you're taking then goes directly into your bloodstream. It doesn't get broken down the way your doctor expects it to. And so the concentration of that in your blood goes up, 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 because your liver's not breaking it down. So that's enzyme inhibition that can, and again, it's like a crosstalk kind of a thing where this thing is blocking the enzymes. Now this thing's out of control. Enzyme induction inhibition. That increases the half-life for some drugs. And you can imagine what state in the union this was discovered first, right? Florida, where there's all these old people taking all these drugs and all this orange grapefruit juice. So that's where they first found people overdosing on their heart medications because they were taking them with grapefruit. It's not funny. Don't laugh. It's not funny.
Oh, it really is.
Okay, so that's it for. Are there any substances or foods that promote inhibition of the phytochemicals? Yes and no. I think the way you want to look at it is if you flood the enzymes with anything, then they'll get busy and they'll get blocked, and other things can happen. So if you flood it with one chemical and you give it these other phytochemicals, they'll get into your bloodstream more. Grapefruit phytochemicals are probably not dangerous. They might even be healthy. I don't know. So anything that's going to get those liver enzymes busy is going to block metabolism of other things. So there's a major crosstalk thing going there. Okay, does enzyme inhibition play a role in nausea you encounter with certain antibiotics? That's a good question. I don't really know. You know, off the top of my head, not knowing the answer, but making one up because I hate to be a guy who doesn't know the answer, I would say usually your nausea with antibiotics is because of the high concentration of them in your stomach. When you take that pill, it's really high in concentration there, like I talked about in the last class. But, you know, like I say, I'm kind of making that up, so I don't really know. Any other questions? See, it's okay to BS as long as you let everyone know you're BSing. Okay, so we will move on to elimination. Elimination. Here, well, I've got a chat coming up here. Hello. When crosstalk occurs, is it dependent on the amount of liver enzymes, or is it random? Is it dependent on the amount of enzymes? It has more to do with the structure of the chemical you're taking. on a regular basis, you want to figure the amount of liver enzymes is pretty much constant. So this crosstalk happens because this new chemical is changing your liver enzymes, either blocking them or upregulating them. So it has more to do with, it's not random, it's based on the chemicals that you're taking and how they affect your liver. Okay, that makes sense? By the way, Willie, I type much worse than you, so you don't need to apologize. Oops, shoot, what happened? That's not so bad. Actually, that one came out pretty good. My typing is really, really horrible. Okay, so elimination. Okay, I think in my introduction I talked about how some things can come out through your feces. Most things don't. Some things will get caught up in your bile, and then the bile goes out into the intestines and it goes out through the feces or something. A few things get lost that way. Most of your elimination is going to be through the kidneys. And keep in mind too, some drugs will not be eliminated through the kidneys. They'll just be metabolized down into something your cells will use for energy. But of things that are being eliminated from the blood, it happens in the kidneys. So we'll talk a little bit here about kidneys and how they function. Basically, you've got an arteriole here. This is blood coming into your kidneys, and your kidneys get something like 20% of all your blood flow. So a lot of blood is going to your kidneys, and they go into this Bowman's capsule. In this Bowman's capsule, you've got some extremely leaky arteries. Basically, all the fluid in your artery can leak out. Anything smaller than a protein is going to leak out here into this Bowman's capsule. So that's good stuff, it's bad stuff, everything. So basically you've got a dump here. It all comes out and it comes down to this descending loop of Henle. This arteriole is going to come back over here, and in this descending loop of Henle, you will have reabsorption of the things that your body wants, okay? So amino acids and sugars will be dumped out, but then they'll be specifically reabsorbed. So everything that's going to come back into your body will be specifically reabsorbed in these loops. And then there's water balance here too, where I don't remember exactly how it works, but basically the salinity gets higher as you go down this loop of Henle, so water goes out, and then it gets lower again back up this loop, and I think I don't remember exactly, but you're concentrating as well as reabsorbing as you go down and back up this loop of Henle. I learned that in 1982, and I still know it. So when I test you 40 years from now on this stuff, you better still know it. I'll be dead then, don't worry. Okay, so the reason I'm telling you all of this is down here in this loop of Henle, okay, if you've got a polar toxin, it ends up inside the loop, and it's not going to be specifically reabsorbed. It's not something the body wants. So it stays in that tube, and then it'll make its way back out of the tube and into the collecting duct and out into your bladder, and then gets released. But nonpolar things can go anywhere they want. So even though they're getting dumped out here up at this point, they get reabsorbed anywhere along this process because they can go right through the membranes of those vessels. So this is why only polar things are going to be eliminated from your kidney, through your kidneys. And you know, I guess I'm beating a dead horse by telling you this, but that's why your liver is making things more polar, because those two organs are working together. The liver's making the toxin polar, the kidney's getting rid of it. Oops. Okay, let's go back to our notes. Role of the kidneys. Capillary, Bowman caps descending, reabsorbing water, salts, and nutrients. Polar things get reabsorbed. Non-polar things, collecting duct. Anything polar that isn't actively reabsorbed stays in the urine, is eliminated. Metabolized drugs that have been made polar are removed from the blood. Okay, so we've talked about metabolism and we've talked about elimination. Some things won't be metabolized in the liver, they'll just be eliminated through the kidneys. if they're not metabolizable, but they're water soluble enough, the kidneys will get rid of them. I think amphetamines are like this. They're 60% of them are 60% of the amphetamine will be removed unmetabolized, 60 or 40, one or the other, I don't remember. So is that a chat or is that someone coming? I hear ding dong, and I don't know what the heck it means sometimes. All right, well, anyway. All right, where the hell was I?
It means someone came or left.
Oh, okay. I won't take it personally. So metabolism and elimination, you put them together, you get drug clearance. Drug clearance is the product of both, which means it's how long it takes the drug to be removed from the blood. So drug clearance then typically is measured in a half-life, but it depends on the kinetics of how it leaves. So most things are going to be leaved by first order kinetics, which gives you a half-life. Now the half-life here, let me do this real quick. Make it stand out better. Okay, so first order kinetics means whatever your half-life is, half of the chemical in your body will be removed every half-life. So if you've got something at 100 milligrams per mil in your blood, half-life is 12 hours. After 12 hours, half of that will be gone, you're down to 50. After 12 more hours, half of that will be gone, you're down to 25. Another half-life, you're down to 12 and a half, and so on. And that gives you this like hyperbolic decrease in the concentration in your blood, and it asymptotically approaches zero from there. Theoretically, it'll never be out of your body. Of course, it will eventually, but it takes a long time to be totally removed from your body. That's first order kinetics, and most things are going to be removed this way. So it's going on in this like half thing because of your enzymes, basically, right? And when your enzymes are full, they're working as fast as they can. Once half that drug is gone, they're only working half as fast because they only have half as much drug to work on. Half of that goes away. Now they're working as quarter of the fast as they originally were, and on and on and on. So that's how you get your first order kinetics. Some things, very few things, but some things will be removed by zero order kinetics. Alcohol is like this. If you drink alcohol, the half-life of alcohol is 1 hour, which means that it's weird when they say half-life for alcohol because it doesn't really work like a half-life. But basically, the elimination rate, I guess you would say, for alcohol is 1 drink per hour. So if you drank 5 drinks, it would take 5 hours for it to get out. If you drank 2 drinks, it would take 2 hours for it to get out. So it's basically boom, boom. It's a straight line over time. Alcohol is like this. Can't really think of anything else. And actually, the reason for this is because you drink so much alcohol. You college students drink so much alcohol. No, kidding. If you look at like how much alcohol it takes to actually produce its effect, it's a very, it's not a very potent drug. So if you take a shot glass of alcohol, that's, it's about, what is a shot glass? Maybe let's say 40 mils. You're going to drink 40 mils of alcohol. It's only 50% strong whiskey, 50% alcohol, the other's water. So now you're down to 20 milligrams or 20 mils, which is about 20 grams. So you're taking 20 grams of alcohol into your body for 1 shot. If you take a 20 milligram tablet of Valium, it's even stronger than that 20 grams because it's a very specific drug. It binds to specific receptors and has an effect. Alcohol is a lot less potent. So that's why, you take so much of it, you overwhelm the enzymes. And so they're working as fast as they can. So you get this zero order kinetics as it comes down. Now, if you can look at alcohol in your body, like way past you when you can feel it, like way down here, you'd get that asymptotic approach to zero again because the enzymes are no longer saturated. But because you take so much alcohol, it looks like a zero order elimination rate. Okay. And I like this animation. It's pretty simple and dumb. Let's stop our share. What I want? Nope. And- Okay, can you see this animation? So this is kind of a simple animation showing you time course of let me read it so I don't say it wrong. Time course of drug effect. The route of administration is greatly affected by the peak of the drug. Okay, so all right. Before I talk about this, let me go back to my notes and see if I finished. Half-life, 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 half-life. Okay. All right, before I get to this thing, let me talk about things that affect half-life. Okay, so what's going to affect the half-life? A lot of things will affect this. And some of them depend on the drug. Some of them depend on the person. So variabilities in terms of the drug is polarity. We keep talking about polarity. It's really important. If it's not circulating in your blood because it's stored in that second compartment, it's not going to be metabolized. And so that gives it a really long half-life. It's only that little bit that's leaching out as you reach equilibrium, as you eliminate that non-polar drug from your body, a little bit more leaches out, a little bit more leaches out. So if it's non-polar, you're going to have a long half-life because you can only metabolize what's in the blood. And also, the liver has different ability to metabolize different drugs. Some drugs are like drugs that are esters, like cocaine, are really easy to break down. Esterases are everywhere. So they get metabolized quickly. Other things are just not things that your liver is used to looking at and doesn't really know how to work with. So some drugs have longer half-lives and some drugs have lower half-lives. Now, when you look at the same drug across different people, different people will have different levels of enzymes, different isoforms of enzymes. Like I say, this cytochrome P450 is a whole bunch of different enzymes, and it's all genetics. Pharmacogenetics is another big field now, where everyone's got different enzymes for metabolism, so the amount of metabolism of the drug in different people will vary. Also, from one person to another, in terms of age and state of your health, that'll make a big difference. Sick people with bad livers don't metabolize well, and so drugs build up in their system. That can be really dangerous. Young, healthy people like you, who I envy, frankly, have got really strong livers, really good livers, so things get broken down pretty easily. So that'll make a big difference. Body composition, again, talking about a non-polar drug. If you've got a larger fatty compartment, you're going to increase the half-life of a non-polar drug. Distribution of non-polar drugs to fatty tissue delays removal. Non-polar drugs stay in fatty tissue. Okay, I'm beating that dead horse. And also, kidney function is going to vary between different people, especially with age. Older people, kidneys don't work as well because they're getting old and beat up. So that can cause drugs to build up in the system. And also other physiological things like, age, I've talked about gender. There's a difference in body composition between males and females, and body size. Hormones make a difference. Big differences between different genders that can affect metabolism of drugs. I always get nervous when I use the term gender because I'm old and the term gender has changed meaning so much in the last 20, 30 years. Please don't be upset with me if I use it the wrong way. I'm not really sure, how to use it anymore. I used to say sex, and then people are like if I'm having sex, the drugs will get broken down more. No, definitely not. It's nothing to do with that. Oh, here's a funny meme from the internet. I don't know if you've heard this one. Someone posted somewhere, what are the best drugs to be on if you're having sex? And someone posted birth control. Okay, pregnancy. That's another thing that can affect drug metabolism. And that's about it for this chapter. What time is it? 4:19? 4:19. That gives us quite a bit of time to talk. So I've got this one animation which is a little bit boring, but I like it. I like it. Okay, here we are. So, this is important. This is important. So how long is the drug going to stay in an effective dose in your body? Has to do with both absorption and metabolism elimination, okay, clearance. Because the slower, I said this before, but the slower it takes for a drug to get into your body, the longer it's going to be in your body. Let's watch that happen. So that's IV injection. Can I pause this? I cannot, no. I'll talk about it afterward. Oral and oral extended release. Okay, so let's talk about this. This is kind of, oh, damn it. Damn it to hell. Is there any way to pause this? Actually, I think I might have a still figure of this. Okay. This is kind of an important topic too. All right, IV. IV comes in really, really fast, right? It's basically very little absorption time. So as soon as it goes in your body, it's maxed out in your body. Now there's no more absorption. All there is elimination. So it gets eliminated the fastest because there's no time for absorption. Okay, IM takes a little time for absorption, but as soon as it peaks, then you're talking about only elimination. Okay, so let's just go through all of them first. Okay, here is oral. Oral, as we said, takes a long time to get into your system. It then peaks, and then after the peak, you get into a purely elimination phase, and then it comes out. So in terms of how long it's in your body, the oral will be in your body the longest, and the IV will be in your body the shortest. And that's, oops, what? What are you doing? Oh, I must have hit that by accident. Sorry. So two things to look at here. If we're talking about absorbing the same dose of the drug, IV will give you the highest peak, right? And that's because all the drug gets in your body at once and it peaks in your blood. Oral will give you the lowest peak. And the reason for that is in here, when you're absorbing, right, it's coming in slowly. As you're absorbing, you're also eliminating, right? Elimination never stops. So as it comes into your body, it's being eliminated. So that's going to depress your peak. By the time you're through absorbing, you've already eliminated, 50% of the drug. And then here on the downslope over here, you're still absorbing some of the drug as you're eliminating, and eventually all the drug will be absorbed and you got only elimination over here. So these processes are happening at the same time in oral administration, whereas with IV, you've only got your absorption's immediate and you've only got elimination going on. So it's going to peak the highest and last the shortest for that reason. Here, this blue line is your oral extended release, controlled release. So this is a really neat way of doing drugs today. It's probably 20 years old, or maybe less, I'm not sure. But basically, if you take a pill where it's just the drug, it goes into your stomach, it dissolves, it gets past your intestine, and it gets absorbed. And then, again, you've got this dynamic here where as it's being absorbed, it's eliminated, it only lasts for so long. What the extended release does is they formulate that thing, they package it up in such a way that it's not going to dissolve in your stomach and be totally released. It's going to dissolve, it's going to sit in your stomach, get past your intestine, and slowly break down as it goes down your intestine. So as it's breaking down, it's releasing drug. So you're getting a constant release over a long period of time. So this is the difference between oxycodone and OxyContin. Oxycodone, I think, is Percocet. I'm not sure. But OxyContin then is like the exact same drug, but in a higher concentration, 4 times as high, extended release formulation. So the idea is if you're taking the Percocet, you need one every 4 hours because it only lasts about 4 hours. You take the OxyContin, you've got a 4 times dose, you take one every 12 hours, right? But it's being really slowly over time instead of all at once. And where a lot of people got themselves in trouble with OxyContin back in the day was they would take that OxyContin, crush it up, destroy that extended release thing, and now you're getting a 4 times dose all at once, So they changed them so you can't do that anymore. They've changed the formulation so you can't just grind it up and destroy it. But that's what extended release is. It's formulated so that it releases slowly in your intestine over time. Okay. What else do we got? Okay. Okay, any questions? Comments? Nobody's got nothing for me. Okay, that's about it for this chapter. Okay, that's chapter 1. I'm hoping that you're reading the book because I go into a lot more detail and stuff in the book, and it's extremely well written, if you don't mind my saying. No, but I think it's interesting anyway. So I would say make sure you're reading that. That's all I have for today. Any questions? No questions, nothing. Celine, what are you drinking there? Day drinking in the COVID? I understand.
No, I'm drinking coffee.
Ah,
Listen, when you wake up at 4:30, you let me know how you feel.
okay. I'd be drinking Irish coffee this time of day. Okay, well, I'm going to let y'all go and not waste any more of your time. I hope you all have a good day, and I'll see you again Monday. Let's see you Monday. Okay. Bye, y'all.